Thank you very much. We're back for the remainder of the morning session. And I want to look out for, this is going to be the mitochondria, integrated stress response, and inflammaging session. In your programs, it indicates that Maria Cuervo was to be the moderator. Unfortunately, she's had a family emergency and she's unable to be with us. So we're going to fill in as best we can. And I see Doug Wallace of the Children's Hospital of Philadelphia and Hyungdun Rong. We're ready to go. If you can, Ryu, Doug, would you mind taking the mic and getting us started, please?
So we're really pleased to have you all join us for the mitochondrial dysregulation, integrated stress response, and inflammaging program. We've been coordinated, of course, by Tom, who has been a wonderful host and keeps us on track. And Georgia Zenthio has been a major organizer for this particular session. I'm Doug Wallace from the Children's Hospital of Philadelphia, and we're also going to hear from Hyungdun Ryu, who's a professor of cell biology at NYU Grossman School of Medicine, and Luigi Farici has agreed to be a discussant and talk about inflammaging. Excuse me. So this session is going to start with a discussion about the mitochondria and how mitochondrial regulation might be related to aging, integrated stress response, and inflammation. So the mitochondria is actually a separate life form inside our cells. It's a symbiotic bacteria that formed the symbiosis about 2.5 billion years ago. It still retains its own DNA, which is transcribed into RNA, and that RNA is translated on mitochondrial ribosomes to make 13 polypeptides, and those polypeptides are critical in the mitochondrial energy generating process, oxidative phosphorylation. So the oxidative phosphorylation is the way we burn hydrogen from our food, our glucose and fatty acids primarily. Looking at glucose, it would go through glycolysis to pyruvate. Pyruvate would enter the mitochondria. Pyruvate dehydrogenase would generate acetyl-CoA and drive the tricarboxylic acid cycle. That strips the hydrogens off the hydrocarbons, and the mitochondria then burn the hydrogens by what's called the electron transport chain, complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then reduces an atom of oxygen to a molecule of water by the 2 electrons. The energy is used to pump protons out to create an electrochemical gradient that's negative and alkaline on the inside of the mitochondrial matrix and acid and positive on the outside. And that potential energy is used by the ATP synthase to drive the synthesis of ATP from ADP and phosphate to make ATP, which is exchanged across the inner membrane by the adenine nucleotide translocator, where it goes out to do work by ATP hydrolysis. Now, different people in the world have different efficiencies at pumping protons out and converting them to ATP. Those people that are very efficient at burning hydrogen to make ATP are called tightly coupled, and they will, in fact, generate the maximum amount of ATP for the minimum amount of calories. The people that are less efficient at pumping protons or converting them to ATP will have to burn more calories for the same amount of ATP. Now, a calorie is a unit of heat, so the more calories you burn in your body, the more heat you generate, and it's this coupling efficiency that allows us to control our body temperature relative to our work demands. The mitochondria also makes reactive oxygen species, and it also has a self-destruct system, the permeability transition pore, that allows the cell to be degraded from inside out when it becomes sufficiently damaged by oxidative stress, elevated calcium, or reduction in energy production. So the mitochondria generates about 90% of your energy, regulates redox balance, generates reactive oxygen species, regulates calcium, regulates cell death through apoptosis, and as you'll see, regulates the epigenome through mitochondrial TCA cycle intermediates. Now, the mitochondrial genome is now only 60,569 base pairs because about 1 to 2,000 of the original genes are now coded in the nucleus. So these 2 genomes must be coordinately regulated to generate the OXPHOS complexes. Complex 1 being made of 45 proteins, 7 from the mitochondrial DNA, complex 3, 11 proteins from one mitochondrial DNA, complex 4, 13 proteins, 3 from the mitochondrial DNA, and complex 5, 17 proteins, 2 from the mitochondrial DNA. So really, the nucleus in the mitochondria must be coordinately regulated to assemble this complex organ, this complex organelle, which is really a bacterium from 2 distant genomes. So the mitochondrial DNA, as I said, was 16,569 base pairs. It's a circular molecule from the original bacterium, codes for regulatory region, small and large ribosomal RNA, 22 transfer RNAs that punctuate the genes, ND1, 2, 3, 4, L4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, and ATPase 6 and 8 for complex 5. Now, the mitochondrial DNA is exclusively maternally inherited, being transmitted from mother to all of her children and her daughters to their children, but males' mitochondria are seen as foreign and are destroyed before they enter the egg. The mitochondria has thousands of copies, and they're constantly replicating in your cells, so you accumulate mutant mitochondrial DNAs, and we call this mixed cytoplasm heteroplasmic. If the cell divided down the middle, both daughter cells would have some mutant and normal, but if it divided this way, this cell would have only normal mitochondrial DNAs, and this cell would have mutant. So over time, we accumulate these mutations with age, and they segregate during cell replication so that you can have a distribution of mitochondrial DNA genotypes throughout the individual's body by this random segregation of mutant and normal mitochondrial DNAs. And when the amount of mitochondrial DNA mutations gets so high that there's insufficient energy for that organ to function, that crosses the bioenergetic threshold, and that creates a clinical phenotype. And the organs with the highest energy demand are the brain, which is 2% of your body weight, but uses 20% of all of your body's energy, followed by the heart, muscle, kidney, and endocrine systems, which are the common organs commonly affected with aging. The mitochondrial DNA, because it's exclusively maternally inherited and has a high mutation rate, it's allowed our ancestors to adapt to different environments as they migrated around the world. So the first mitochondrial DNAs we find in sub-Saharan Africa, we call that L0. L1 and L2 are 2 different pygmy lineages. This is 90,000 years old. This is 30,000 years old. L3 are sub-Saharan Africans, primarily Bantu-derived people. In Ethiopia, 2 mitochondrial DNAs arose, M and N, and they were the only mitochondrial DNAs to leave Africa and colonize the world. M moved along the tropics all the way down to Australia, much later moving into the temperate zone to give you a series of mitochondrial DNAs, including C, D, and a series of M. But N moved directly into the temperate zone to give lineages like H, J, T, U, U, K, V, and then moved laterally to mix with the M lineage mitochondrial DNAs in Asia. And then in Siberia, only 4 mitochondrial DNAs, 2 from Europe, 2 from N, and 2 from M, crossed the Bering Land Bridge and colonized the Americas. And the reason for this geographical distribution of the mitochondrial DNA is because the mitochondrial DNA variation is affecting the coupling efficiency of our mitochondrial DNA. And those individuals that live in Africa would want tightly coupled and very efficient mitochondria to run away from lions. But up here where the lions froze to death, you would want to uncouple your mitochondria by mutations, so you would burn more calories for the same amount of ATP, but maintaining a high core body temperature to resist cold. So the mitochondrial DNA then has been very important in a wide variety of adaptations to environments, as you will see. So there are 3 classes of mitochondrial DNA mutations. Mutations that occur along the maternal lineage, like this mutation in the tRNA leucine at 3243, that's the nucleotide position. When it is heteroplasmic at about 30% heteroplasmic, it gives diabetes and autism. When it's 80% heteroplasmic, it gives neurological and muscle disease. When it's 100% mutant, it kills you as an infant with Lee syndrome. Or mutation in the tRNA leucine gene at 11778. If you inherit that from your mother, that is pure mutant homoplasmic, you'll be fine until midlife, then you'll lose vision in one eye, and in the other, Leber's hereditary optic neuropathy. There are now 1,000 putative pathogenic mitochondrial DNA mutations. In our website, MitoMap, you can find out all about them, about 100 of which we've been able to validate. In addition, there are ancient polymorphisms. This variant is found in 3/4 of sub-Saharan Africans, and it defines these lineages. This lineage defines half of the European lineages, A, C, A, B, C, and D arose in Asia. They crossed the Bering Land Bridge, colonized the Americas. And then finally, we accumulate somatic mutations with age, and that's one factor in the aging clock, as a follow-up from Dr. Luigi Ferrucci's presentations. So just as an example, we can look at mitochondrial lineages called haplogroups or groups of haplotypes, and we can see that they can relate to metabolic syndrome, to autism, and to longevity. So here we can see that these mitochondrial lineages are at a decreased risk for type 2 diabetes. These mitochondrial lineages are at increased risk with odds ratios of 2 to 3 of hypertension and of obesity. These lineages are at increased risk for autism and with odds ratios of about 2. And as you probably know, males are 4 times more likely to get autism than females. So these mitochondrial lineages are at least half as important as gender in risk for autism. And this is about 55% of the European populations. The age-related effect of this is seen if you now look at siblings of pedigrees with autism. The affected child has more somatic mutations than the unaffected child. So what that means is this predisposes to autism, but then the somatic mutations crosses the expression threshold and gives you the phenotype. And finally, there are now clear evidence that mitochondrial haplogroups J and U3 are at increased longevity in Europeans, and haplogroup T increases longevity in Asians. So our basic idea then is that we are born with a particular energetic capacity, and then as we age, we accumulate somatic mutations. They erode our energetics until we cross these expression thresholds, and that gives us age-related diseases. And you can see that the somatic mitochondrial DNA mutations accumulate with age in both, in this case, mice and in humans. And if we now make a mouse with an antioxidant enzyme and mitochondrially targeted catalase into the mitochondria, we can increase the longevity by 20%, both medium and maximum, and reduce the mitochondrial DNA somatic mutation by half. So that implies that mitochondrial somatic mutations are an important factor in the aging clock. So basically, if we put energetics in the middle of medicine, we can then explain many of the characteristics of the age-related diseases. So mitochondrial oxphos can be affected by mutations in the nuclear DNA, by changes in the epigenome, by ancient adaptive mitochondrial DNA polymorphisms or recent deleterious mutations, by changes in diet, by how you use your energy, or whether you are exposed to toxins like smoking or rotenone. And if you impair mitochondrial function, you will increase mitochondrial ROS production. That inactivates the proofreading function of mitochondrial DNA polymerase, results in the accumulation of somatic mutations over time, and that gives you the aging clock and explains for the delayed onset and progressive course of disease. The organs that are affected are the most energetic, the brain, giving you the neuropsychiatric syndromes, the heart, muscle, and the renal system. And if you impair mitochondrial function, you accumulate substrates, fatty acids, and carbohydrates, and that's metabolic syndrome. Mitochondria then is specifically related to stress because it's a bacterium. If it's released in the bloodstream, it activates the inflammasome and the interferon pathways through the C-gas sting and the inflammasome pathways, hence a relationship between aging and inflammation. And then finally, all of these diseases have an inflammation component, just as aging does. And finally, cancer and viral infection are very important in energy. So just taking as an example, I don't know how to get rid of this. I'm sorry, we're going to now talk about a particular family with a mitochondrial DNA mutation that affects a tRNA in the mitochondrial DNA at nucleotide position 3243. This mutation was found in the mother, about 70% of the mutation mutant. She has lactic acidosis and growth retardation. These are the children we could examine. They have lactic acidosis, growth retardation, progressive dementia, stroke-like episodes, hypertrophic cardiomyopathy, cardiac conduction defects. oxidative muscle fibers are degenerating, the glycolytic muscle fibers are fine, and these young men and women all died of either heart failure or deep seizures. This pedigree had 70% mutant, giving you muscle, heart, and neurological diseases, but the same mutation at 10 to 30% mutant will give you type 1 or type 2 diabetes or autism. At 100% mutant will kill you as an infant with Lee syndrome. So we were curious about how the same mutation could give you the different phenotypes. So we made what we call hybrids, where we have cell lines with the same nucleus, but with different percentages of the known mutant. And then we did RNA-seq on these cell lines, and we plotted the principal component analysis of all the RNA transcripts. So this turns out to be the principal component analysis of the normal mitochondrial DNA with no mutant. This is with 20 to 30% mutant, equivalent to autism and diabetes. This is 50 to 90% mutant, which is associated with neurodegenerative disease. This is 100% mutant with Lee syndrome and perinatal lethality. And this is a cell line with no mitochondrial DNA at all. And these are the transcription factors that are up or down regulated to give this result: diabetes, neuromuscular disease, lethal childhood disease, and a cell with no mitochondrial DNA. So how did they I'll put this down there. So how could it be that a mitochondrial DNA mutation could result in changes in the nucleus? Well, if you mutate the mitochondrial DNA, you affect the electron transport chain. That will diminish the ATP production. But more interestingly, it will mean that the NADH cannot be oxidized back to NAD, so that this part of the pathway is inhibited. Now, the tricarboxylic acid cycle the tricarboxylic acid cycle requires NAD to move forward as part of the oxidation process, with the NADH then being oxidized by the electron transport chain. But if NADH builds up, then NAD is limited, and that stalls the electron transport chain. So pyruvate goes into this mitochondria. There, it's converted to acetyl-CoA by pyruvate dehydrogenase. It interacts with oxaloacetate to give citrate. Citrate goes out through the citrate carrier, and citrate is cleaved by the ATP citrate lyase to give you acetyl-CoA. And acetyl-CoA, we've now shown, is the main substrate for all the acetylation of the histones and all the sirtuin proteins that interact with the sirtuins. The mitochondria also regulates the single carbon metabolism. That with ATP gives you S-adenosylmethionine, and that is responsible for methylating both the DNA and the histones. Alpha-ketoglutarate is the key cofactor for the dioxygenases, which regulate HIF-1 alpha, and Jamangi dioxygenase and TET dioxygenases, which are the demethylases for the histones and for DNA. And succinate is an inhibitor of these dioxygenase pathways. So what that means is that the mitochondrial substrates are in fact regulating the epigenome directly. So if we now look at cell lines with only 0% mutant mitochondrial DNAs or 100% mutant mitochondrial DNAs, and we look at all the histone acetylation, we can see that there's a diametrically opposed situation where the two genotypes directly change histone acetylations. If we look at all the metabolites of the mitochondria against all the mitochondrial genotypes, against all the histone modifications, we see 150 discrete mitochondrial histone modifications that are involved in regulating the epigenome relative to energetics. So the mitochondria then directly regulates energetics and therefore epigenomic regulation within the context of the age-related mitochondrial diseases. So how can we test this idea directly? We started looking at an environmental challenge, and that is the infection of the mitochondria of the cell with SARS-CoV-2. SARS-CoV-2 is a single-stranded plus-strand RNA virus. It makes these polypeptides for its replication and cell regulation. They get cleaved into non-essential proteins, and it codes for the S protein, E, M, and N, which are important core proteins for the virion, and it also codes for a whole series of open reading frames that turn out to be virulence factors. If we now treat a cell with a virus, and the mitochondria fragments into fragments, and the mitochondria adjacent to the replicating sites of the virus become highly pyknotic with abnormal cristae. This is the cristae of a non-infected cell. So what's happening in oxidative phosphorylation is that the enzymes are very, very complicated, so they have to be assembled in modules. So there's an ND4 module, that's a mitochondrial DNA-based module, ND2 mitochondrial DNA-based module, ND1 CoQ based module, a CoQ module, and an N module, and these are the structural proteins together with assembly proteins shown in red that ultimately give rise to your major complex. The same is true for complex 3, where we have structural proteins, assembly proteins to give you the final complex, and likewise for complex 4, where each of the 3 major units are founded by a mitochondrial DNA encoded subunit CO1, CO2, and CO3. Okay, so then what happens when we look at the viral infection of the cell? So now what we're doing is we're looking at autopsies from COVID-19 patients. We looked at 40 autopsies, and we found that there was a marked differential expression of OXPHOS genes and the assembly proteins for their assembly. And what is most striking is that the heart was the most severely affected, followed by the kidney and the liver, and then the lung in the opposite direction. So this just shows the nasal pharyngeal sample, and we have OXPHOS genes are completely downregulated. By the time you get to autopsy, the virus is cleared and the mitochondrial respiration is upregulated, but in the heart, it stays down. In the kidney, it's down the liver and the lymph node. So this just shows all of the nuclear encoded OXPHOS genes, these being the mitochondrial DNA encoded ones, are downregulated in complex 1, complex 2, complex 3, complex 4, complex 5, and so on. So there's a coordinate inhibition of mitochondrial transcription, because these are messenger RNAs, transcription of the OXPHOS genes. And what's really surprising about that is that the virus only inhibits certain groups of proteins that are involved in assembling the modules. So this is one module, and all those genes are downregulated. Another module, all those genes are downregulated, and so on. So if you look at the relationship between the assembly genes in tan and the structural genes in brown, you can see that often the structural genes are regulated by the virus in one way, and then the host responds by upregulating the assembly genes in the opposite direction. We see that over and over again. So we have a battle between the virus downregulating mitochondrial gene transcription in the nucleus and the host trying to compensate by upregulating the uninhibited genes. And also, a major point, the virus activates the mitochondria to generate in a system to activate the inflammasome using mitochondrial oxidase mitochondrial DNA replication that is exported through the permeability transition pore and binds to the inflammasome. So then what we have, we're getting close to the end, is we have a situation where the double-stranded virus can activate a system for viral and host protein messenger RNA cleavage through the 2,5' oligonucleotide synthetases, but the virus can bind also to the MDA5 protein that activates the mitochondrial MAVs, and that activates the interferon pathway. But also the virus can activate in the ER the PYR protein that phosphorylates the PYR protein, that phosphorylates the elongation factor. That phosphorylation of the elongation factor stalls the protein synthesis system that allows the activation of these transcription factors, ATF4 and 5. They upregulate mitochondrial supercomplex formation, turn on the genes FGF21 and GDF15. And by the way, the last speaker on the last session showed that GDF15 was upregulated in all the aging systems. And that is the integrated stress response that's very important in the effect of the virus and in aging. So basically then, if we look now at all the genes that are involved in the HIF1 alpha pathway, which regulates glycolysis, mitochondrial oxphos inhibition increases ROS production, that activates HIF1 alpha, and that activates the pseudo-hypoxic state. And what you see is all the genes for HIF1 alpha are generally upregulated. So we see a marked upregulation of the pseudo-hypoxic state, HIF1 alpha, as oxphos is downregulated. We also see the differential regulation of the mTOR system that provides substrates to the mitochondria. It's downregulated in the heart where the heart is failing, but in the lung where the oxphos is going up, we see the upregulation of mTOR. But most interestingly, in all the organs, we see a coordinate upregulation of the genes of the integrated stress response, the system trying to cope with the viral transcriptional inhibition. So finally then, we can see that as the SARS-CoV-2 infection progresses from acute to early to mid to late stage to late, that initially oxphos is only moderately affected. In an early stage, it's downregulated, and then by the time you get to the late stage, in the lung, it's upregulated again to compensate for that, but in the heart and all the other organs, it's downregulated, resulting in death. HIF1 alpha is upregulated in the whole system to compensate for the oxphos downregulation. mTOR is generally upregulated except for in the heart, and the integrated stress response is coordinately upregulated. So we see a direct interaction between mitochondrial dysfunction, integrated stress response, inflammation, and inflammaging. So finally, we think that the mitochondria is a major mediator of nuclear and cytoplasmic gene expression and environmental challenges. When you inhibit mitochondrial function, you alter the mitochondrial metabolites. They act as signal transduction systems for the epigenome, which then changes the gene expression of both the nucleus and the mitochondria to maintain energetic homeostasis and health. But if any of these gets too severe, the system cannot compensate by the epigenome, and you get a decline and death. And these are the wonderful people that did the work. These are the folks that did the work on the epigenomic studies. These are the people that worked on some of the age-related diseases, including the autism, and this is a very large team that does all the work on COVID-19. So that's then that part of the discussion, and I will stop sharing and then turn the program over to my colleague, who is Huang Don Rio, and maybe he can tell us a little bit about autophagy and mitophagy.
So thank you for having me here. And I think Dr. Doug Wallace's presentation was, he covered a lot, and including the integrated stress response, so I can pick it up from there. And I have a comparatively shorter presentation, but I'll try to stick on the conceptual aspects of the integrated stress response. And if there are any questions along those lines, then please feel free to interrupt. Okay. So let's start with the term, the integrated stress response. So why is this signaling pathway called the integrated stress response? And part of the reason is because the signaling pathway that I'm about to tell you is activated by many different types of stress. So here are listed just 4 different kinds. Dr. Wallace talked about viral RNAs. If there are double-stranded RNAs, then they can bind and activate a stress-activated kinase called PKR. This was one of the first kinases that were identified in the integrated stress response. And then subsequently, these are all very old discoveries, but my old colleague here, David Ron, who used to be at NYU and currently at the University of Cambridge, identified a protein, a kinase called PERK, and it really stands for PKR-like endoplasmic reticulum kinase. And this turns out to be a stress-activated kinase that specifically responds to misfolded proteins in the endoplasmic reticulum. way before that, now really a leader in this field, Alan Hinnebusch at the NIH, had studying from the 1980s how yeast cells respond to amino acid deprivation. And he had been studying an amino acid deprivation activated kinase called GCN2. And also similarly, this is HRI is an enzyme, a kinase that had been studied for a really long time. It stands for heme responsive kinase. It's heme deficiency activates this, but very recently people have identified that there is a mitochondrial factor called DELE1. And when that's released from the mitochondria due to mitochondrial stress, then that binds to HRI to activate this kinase. So these set of kinases, they really respond to many, many different types of stress. But at a certain point, the scientists decided to use the term the integrated stress response, because these different kinases, they ultimately activate a common and integrated response by phosphorylating a common target. And that is a translational initiation factor called EIF2. And it's the alpha subunit of EIF2. EIF2 has 3 subunits, alpha, beta, gamma. And it's phosphorylates EIF2 alpha. Now, I'll get into this a little more in detail, but EIF2 alpha is a translation initiation factor. It plays an important role bringing initiator methionine to ribosomes. So without this, there's going to be a real problem in initiating general mRNA translation. And these kinases basically phosphorylate and inhibit this protein. So how would that help with stress response? For example, amino acid deprivation. If cells don't have enough amino acids, then it might help by reducing overall protein synthesis. So that's one way by which this pathway is thought to help cells suffering from amino acid deprivation. What about endoplasmic reticulum stress? Well, the ER is most often suffering because it's having a hard time folding nascent peptides. There are more nascent peptides or misfolded proteins fold in comparison with the protein's chaperone capacity. So under those circumstances, if there's reduction in overall protein synthesis as a result of EIF2 alpha phosphorylation, they might provide some temporary relief. But the pathway doesn't stop there. Now, general mRNA translation is suppressed, but something very strange happens, which is now a transcription factor, and most famously, the mRNA encoding a transcription factor of ATF4. This translation is paradoxically stimulated when all the other general mRNA translation is suppressed. And I'll get to that particular mechanism in a minute or two. And once ATF4 is now synthesized, because it's a transcription factor, it would induce gene transcription. And many of its ATF4 target genes are various stress response genes, just to name a few to tie in with Dr. Wallace's presentation. For example, mitochondrial stress. Okay, so he mentioned, among other things, folate metabolism, 1-carbon metabolism. Many of the 1-carbon metabolism genes are under the control of ATF4. They're induced by ATF4. And there was a paper by one of our recent colleagues several years ago. 1-carbon metabolism ultimately helps generate this thing called 4-mil THF, which is a raw material for 4-mil methionine, which is important for mitochondrial protein synthesis. So this pathway is going to affect mitochondrial metabolism in a major way. Amino acid deprivation, ATF4 induces many non-essential amino acid biosynthesis and also import. So that's going to help with amino acid deprivation by producing more pools of amino acids. And it also induces many endoplasmic reticulum quality control genes that will help with endoplasmic reticulum stress. Okay, so now, what is there more to study? And my lab has been really interested in a conceptual problem for the last several years. So as I introduced earlier, this pathway involves mRNA translational suppression with transcription. Now, if you think about it, okay, ATF4 has induced lots of mRNAs that if they're translated, they're going to help cells. But there's a translational block. So we have to, a missing component, a very important missing component is how do those mRNA stress responsive transcripts actually get translated when there's a general suppression of mRNA translation in cells. And one of the components that we've been focusing on in the last several years is a protein called 4EBP. So let me introduce that protein first. So most cellular mRNA translation is thought to occur through cap-dependent translation. As you know, mRNAs are a little different from tRNAs or rRNAs in that their 5' end, there's a methylguanosine group in a very peculiar conformation, and that's generally referred to as the 5' cap. And that's important for mRNAs because that gets recognized by an important translation initiation factor called EIF4E. And EIF4E's role is once it binds to the caps, it recruits the small ribosomal subunit so that now this mRNA can get translated. And 4EBP, which I'm about to highlight in the context of this integrated stress response, is a EIF4E binding protein, 4E binding protein, and it inhibits this general mRNA translation. So, okay, so 4EBP sounds like a very generic protein, perhaps a housekeeping protein. So what does that have to do with metabolism and aging, which is the focus of this meeting? Surprisingly, it's very relevant. So let me just show you two different examples. So we do most of our experiments in the fruit fly, but in relation to TOR signaling, many of you would be familiar. mTOR is something that responds to nutrients and it controls growth. And if you inhibit mTOR, for example, the popular drug nowadays is rapamycin, and in experimental organisms, people think that rapamycin can really, it reduces growth, but it enhances stress resistance and it promotes longevity in experimental animals. And in fact, there are public news media articles about physicians actually prescribing rapamycin to prolong lifespan of their patients. Whether it's legal or not, it's disputable. Many scientists think it's a little dangerous. But how does mTOR do its job once it's activated? One of its major target is 4EBP, the protein that inhibits EIF4E that I just told you about. And if 4EBP is inhibited by TOR, then now EIF4E can promote mRNA translation. It leads to enhanced growth and fertility. If you inhibit 4EBP and now, TOR, and 4EBP is therefore active because it can't be phosphorylated, then what people find is that it enhances stress resistance, and at least in experimental organisms, it enhances, prolongs lifespan. And we got interested in this particular problem because years ago, many scientists, independent groups, have reported that there's another mechanism to regulate 4EBP, and that is, for example, limitation of amino acids, proteins in the diet. And 4EBP transcription is enhanced under those conditions, and those conditions enhance stress resistance and prolongs lifespan in the fruit fly. And these are all independent laboratories, and they came to the same conclusion. And what they didn't figure out back then in their studies was the mediating signaling pathway that leads to the transcriptional induction of 4-EBP under these conditions. And we got into this particular problem by showing that it's the integrated stress response that mediates amino acid deprivation, induced 4-EBP transcription. And we showed that the upstream kinase GCN2 that mediates the integrated stress response, that in fact affects the longevity of the fly under limited amino acid conditions, and that ATF4 is really directly activating this translation initiation factor. Okay, so we started out with introducing the idea that virtually all, well, not all, but most cellular mRNA translation occurs through this basic mechanism for EIF4E mediating translation. And if 4-EBP inhibits every single mRNA translation, then it shouldn't really have this specific effect in stress resistance and lifespan. But what we're seeing over and over again in my lab, and also we can reinterpret many of the papers published earlier by other groups, is that not all mRNA, it is certainly true that most cellular mRNAs undergo EIF4E dependent translation, but not all. So when 4-EBP is induced under stress, and ATF4 now induces many other stress response transcripts, what we're seeing is many of the ATF4 targets actually have an unconventional mechanism to recruit the ribosome. And this is perhaps why 4-EBP enhances stress resistance. It would inhibit growth-promoting protein expression, but it does not inhibit many of the stress-responsive transcripts that are actually induced by ATF4 during the integrated stress response. And we have published several mechanisms, for example, stress-responsive transcripts having internal ribosome entry sites. Those are 5' UTR sequences that recruit ribosomes independent of EIF4E and the 5' cap, and there are probably additional mechanisms. Just to highlight that that's not the end of the story, I didn't even touch upon this idea. Well, how does ATF4 get translated when EIF2 alpha is phosphorylated and inhibited? Under these conditions now, ribosomes can't get initiator methionine tRNAs. But under those conditions, ATF4 paradoxically gets synthesized. And recently we found that there is another EIF2-like protein, and we didn't name it, but other biochemists have given the name EIF2D, not because it has sequence similarity, but it has functional similarity in the test tube. It can bring initiator methionine tRNAs to ribosomes. And we find that EIF2D and other proteins with similar activities are actually required for ATF4 synthesis in stressed cells. So these are some of the mechanisms by which stressed cells bypass conventional translation initiation mechanisms to induce stress-responsive transcripts, and we think that it makes sense that stress-responsive transcripts will use a lot of unconventional translation initiation mechanisms to ensure their proper synthesis to help cells maintain homeostasis. Now, I just want to cover one more slide, and that is the idea that ATF4 is not the only transcription factor involved in the integrated stress response. So ATF4 has the ability to bypass inhibition, translation inhibition occurring through EIF2 alpha phosphorylation. Recently, we identified another transcription factor in the fly, and that's a transcription factor called XRP1. And the name doesn't really matter, but it has this 5' UTR with small upstream open readings. And Alan Hinnebusch had originally characterized an yeast gene that can bypass EIF2 alpha phosphorylation mediated translation suppression and that is the yeast gene called GCN4. And he did extensive work to show that the small upstream open reading frames basically allow these transcription factors to evade suppression by EIF2 alpha. And subsequent researchers studying mammalian genes, human genes, have identified ATF4 based on this type of a 5' UTR structure. And what we're seeing is that there are additional transcription factors with these types of unconventional 5' UTR that allow them to be paradoxically induced in their translation when the integrated stress response is active. And it's not just the XRP1 that we've identified in the fly, but there are now several examples that have been identified in mammalian cells. So the integrated stress response should not be equated with just ATF4. It is more like the induction of several transcripts that have these unconventional 5' UTR that allow stress responsive gene induction. So with that, I'll conclude my presentation. This is my lab photo, and many of these people, including Brian Brown, Finn Roach, and many others who have made important contributions along the way. I'll conclude my presentation here and try to answer any questions that you may have. Thank you.
Yeah, thank you very much, Dr. Waru. We'll start with the questions in the chat. I'd like to go first to your questions. Let's see where we are. Dr. Ferrucci asks, is the set of genes activated by ATF4 always the same independent of what activated the stress response?
Same as those activated by stress response? Was that the question? So there are many different types of stress responses, and the integrated stress response is just one of them. And there are going to be in certain cases, so we're doing lots of gene expression profiling, and another stress response pathway that we study is, that controlled by the insulin signaling pathway, FOXO, the homologue of the famous longevity regulator in C. elegans, DAF16. Many of you may be familiar, and they're activated due to different reasons. FOXO is more sensitive to, for example, insulin signaling. The integrated stress response, as I told you, gets activated by lots of different cues, but not necessarily insulin signaling. But the outcome, there's an overlap. 4-EBP is a common target, very well characterized, but then there are also differences too. And while we were studying the integrated stress response and we were studying XRP1, that study, we initially became interested in XRP1 because we found that these stress conditions that we were studying were activating antioxidant genes. And for a while, we thought, well, antioxidant genes, there are sometimes the literature equates them with NRF2 activation. And that turned out to be a little bit misleading because, yes, NRF2 does activate the antioxidant response, but not always. There are overlaps in downstream targets, not a complete overlap, but there are differences as well.
I expand on this question. This is Luigi. I was wondering how much these mechanisms that are studied mostly in animal model or in model organism translate to human, whether there's differences, and this is really touch on the idea how much the study of this stress response mechanism can be relevant in a translational perspective. I know that it's early, but can you make a comment on that?
Yes. So of course there are clear differences. I mean, I think many of the things that we study in flies probably are not conserved in humans, certainly. But then there are many cases where decades of research turned out that demonstrated that certain pathways are very conserved. And I can just cite two examples. So when it comes to lifespan, of course, one of the earliest discoveries were the roles of insulin signaling from C. elegans. And we saw after the C. elegans studies, people started reporting that flies and mice also, their lifespan can be affected by modulating the insulin signaling response. And then later came insulin downstream. TOR can be regulated downstream of insulin signaling. And now TOR, initially, again, TOR signaling and lifespan started out from yeast and then flies and worms and mice. And the original person who discovered the connection between TOR and yeast lifespan started a clinical trial with dogs in the Seattle area. And now what we're seeing is, if you just Google search like newspaper articles about the TOR signaling pathway and lifespan, we're seeing doctors actually starting to prescribe rapamycin to their patients. So I think it's a little dangerous to jump to a conclusion based on one model organism's findings, but when it comes to when the field matures and scientists generally agree that from yeast, worms, flies, mice, dogs, monkeys, things are conserved, then it'll be very difficult to argue against conservation in humans, in my opinion.
Thank you. That was
Thank
great.
you very much.
So the next question for you, Dr. Ryu, what do you think is the mechanism rationale activating longevity genes when the ISR is activated? Is this due to nutrient deprivation?
That's our current model. And that's, so I remember maybe perhaps like 10 years ago, I used to go to this Ellis Medical Foundation summer symposiums on aging research, and I got to interact with lots of people working on lifespan and model organisms, and the field was ripe with full of various controversies. And people told me, well, if you want to avoid controversy, something that most people agree would extend lifespan in model organisms is a moderate reduction in nutrient intake. Because the way people do experiments is they feed their experimental animals diet that would be maximal for growth and fertility. And what turns out to be the case is what's maximally beneficial for growth and fertility is not beneficial for longevity. That's why a moderate reduction in nutrients is thought to be important in at least model organism lifespan. And generally, I think lots of people believe that that would also apply to humans as well. And those conditions are the conditions that activate the very well-established longevity gene, signaling pathways, such as insulin signaling, TOR signaling, and also the integrated stress response mediated by amino acid deprivation. So that's, I think, has become a very popular frame of thought when it comes to these pathways and lifespan.
Great, thank you very much. Did that answer your question, Dr. Ferrucci?
Yes, absolutely. Absolutely.
Okay.
Thank you very much. That was great.
So question from our sponsor, Dr. Seo. He says, do other viruses hit the mitochondria, or is this one of the reasons why COVID-19 is truly a nefarious outlier? Well, the answer is yes. Other viruses do hit mitochondria, but not all viruses do. And one of the interesting aspects is that the cold virus, that we've done all the parallel experiments with the cold virus and COVID-19, and the cold virus, which lacks most of the open reading frames that SARS-CoV-2 has, does not show many of the same effects on mitochondrial function as does COVID-19. So I think that we're going to have to do one virus at a time and really see how they use their different ways of regulating the system. But of course, inhibiting mitochondrial function is the best way to increase carbon movement into forming nucleic acids, lipids, and so on. And so by inhibiting mitochondria, you increase biogenesis, and many viruses, including cancer cells, all use that approach to increase their biogenesis. Then the next question is, do you see effects upstream of OXPHOS? For example, is AMP dysregulated? The interesting thing was that AMPK transcription was not specifically the main factor in regulating things like mTOR and the integrated stress response. Rather the various other kinases were quite important. AKT kinds of kinases, and that's all in our paper. So you can see the different aspects of these pathways are regulated by different transcription factors. One more question: do you see effects on acetylation status of inflammatory cytokines? I would expect that to see dysregulation of IL-6, IL-1 beta, TGF, TNF alpha, and maybe interferon gamma. And the answer is, by all means, we see major regulatory effect of these different pathways. And interestingly enough, because the mitochondria is now regulating both the CGAS-STING pathway for interferon, but also the inflammasome, that is in fact regulating downstream, for instance, very strong effects on IL-1 beta, on all the interferons, are directly regulated by the mitochondria, generating oxidized mitochondrial DNA that's released into the cytoplasm. The oxidized mitochondrial DNA binds to the inflammasome and activates IL-1 beta, and it binds to CGAS-STING and activates STING, binds to CGAS, activates STING, and that upregulates the interferon pathway. So there's a complete direct link between the mitochondria and regulation of inflammaging. So much coordinate complexity. Can you tell us something about the participation of mitochondria to cellular senescence? Yes, that's of course very active area. There's a strong interaction between the mitochondria and the telomere length. Telomere length is regulated by oxidative stress because the high G content means it gets oxidized, which inhibits DNA replication. And when the telomeres get short enough, then the RNA component of telomerase enters the mitochondria and regulates mitochondrial antioxidant defense systems. So there's a direct interaction between mitochondrial related decline and telomere shortening, and as we just said, with integrated stress response and the inflammasome. So yes, I think these are all integrated, and we're just getting more and more information about what cytokines are being actually generated through mitochondrial dysfunction and how that works. Okay, and we have one more question. Most neurotrophic viruses such as herpes, rabies use mitos and exploit mito trafficking in exonemes. And the answer is yes, mitochondrial trafficking is also a major way that neurotrophic viruses are moved around the body. And of course, if you get shingles, you know that you're going to activate the virus in your, you get the shingle on your skin, but it's because you're innervated through these long axons. So anyway,
Doug, I think that was an answer from Ark to my question about whether other viruses hit mitochondria. But Doug, there are still various questions here. If we could just keep it going a little bit longer.
Oh, okay.
There's Gordon Cutler's question. You see that in the question? And so, do the studies of SARS-CoV-2 effects on mitochondria have any obvious hypotheses on how to treat long COVID? That's a loaded question.
No, not at all. Absolutely. That's where we're going now. We've pretty much done a major study on the actual regulation on mitochondria by the virus and during acute infection. But as what's really interesting is that the heart, the kidney, the liver, even as the virus is being cleared, they've shut down mitochondrial transcription. And the virus also activates an microRNA that specifically affects translation of many of the nuclear encoded mitochondrial proteins. So almost all the symptoms that we see in long COVID are actually classic symptoms that we see in primary mitochondrial disease. And we feel that treating mitochondrial dysfunction will be an important way of treating long COVID. And we're actively developing therapies in that regard, using our mouse and hamster models. And I don't know if Tom, did you want us to go on or stop? I'm not sure. Right. Okay, here's another question.
You're muted, Tom.
Can you tell us something about the participation of mitochondria in cellular? Oh, yeah, we just heard that. Okay.
I think there's a question about long COVID from Gordon Cutler. Do study of SARS-CoV-2 effect on mitochondria have any obvious hypothesis on how to treat long COVID? I think everybody is concentrating on that.
Yeah. So the basic logic is the virus makes proteins that inhibit mitochondrial function. That's the acute infection. I didn't go into that, but that's true. Then it inhibits specific transcription modules that regulate energy production and many other pathways in mitochondria. That inhibits ATP production, but increases mitochondrial ROS. Mitochondrial ROS activates HIF-1 alpha. HIF-1 alpha drives the system to the pseudo-hypoxic state, increasing glycolysis, downregulating mitochondria, and moving all the nutrients into forming NADPH, lipids, and nucleic acids for viral replication. So we're actively working on ways to increase mitochondrial function, to block mitochondrial ROS production, and to regulate HIF-1 alpha expression. And those same ideas can be used also to mitigate the effects of long COVID. Acute infection would be for mitochondrial ROS and HIF-1 alpha regulation, but long COVID is upregulating and reconstituting mitochondrial function. So those different approaches are being used in both our animal model systems and our cultured cell systems.
Luigi, did you have your question answered about what happens to the ISR during physical activity?
Yes. I think that I got my question answered. Yes.
Okay.
I mean, physical activity, just rightly, it's a kind of moderate stress and likely activate stress response. I don't think this has been studied, but I think it would be quite interesting, especially because stress response appear to inhibit translation. But you have exercise where translation is actually incredibly activated. And in that condition, and exercise, of course, is good for you, has a lot of beneficial effect in human and in animal models. So I think that we're dealing with a regulatory system that is quite complex, but very fascinating.
May I also mention something on that regard? So we have created an array of mouse models with both different mitochondrial DNA mutations as well as mutations in nuclear encoded bioenergetic genes. And we've put all of those mouse models through both acute exercise and through long-term aerobic exercise training. And what was surprising about that is that the different mitochondrial genotypes have different responses to a long-term aerobic training. So it's obvious that, or it seems then, that this signaling system is affected in different ways depending on the actual state of the animal at the time. So therefore, we're not sure whether a person with diabetes will have the same effect as a person with neurodegenerative disease. So I think we need more studies on whether aerobic exercise is always the best answer. I'm not saying it's not always good, but whether it's always the best answer is another issue.
Gentlemen, thank you so much. I've just got a signal that we do need to come close out the morning session. So I want to give great thanks to Doug and Hyungdan and Luigi for hanging around.